Greetings, everyone, and welcome to today's webinar titled Utilizing the 6-Base Genome to Investigate the Dynamics of Enhancer DNA Methylation During Cellular Differentiation. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Biomodal. To visit more about our sponsor, visit biomodal.com. I'd now like to welcome our speaker, Dr. Emily Hodges, Assistant Professor of Biochemistry at Vanderbilt University. For a complete biography on Dr. Hodges, please visit the Biography tab at the left of your screen. Now, we encourage you to submit any questions you may have at any point during the presentation today. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you are experiencing any technical difficulties, you can utilize this Q&A box as well, and I will assist you directly. Dr. Hodges, welcome. You may now begin your presentation.
Okay, thank you very much. I'm excited to be here to tell you a little bit about our work and also to tell you about how some of our recent work actually led us to want to do 6-base sequencing to differentiate between methylcytosine and hydroxymethylcytosine at gene enhancers during cellular differentiation. So I'm going to start with a very, very well-established concept, and that is that as cells undergo cellular differentiation, they take on new cell fates, and during these cell fate transitions, the epigenomes of these cells change to adopt their new programs of transcriptional activity. And this process involves multiple layers of the epigenome that are dynamically modified. This includes chromatin that's made up of nucleosomes that repositions around gene regulatory elements to restrict or permit access to transcription factors. The nucleosomes themselves are made up of histones that have amino acid tails that are post-translationally modified in a combination of ways that reflect the local genome activity of these gene regulatory elements. And then there's DNA methylation, which itself is pretty special because this modification happens directly to DNA in the context of CG dinucleotides. And the textbook understanding around DNA methylation is that if you have DNA methylation at a gene regulatory element like an enhancer or promoter, it's transcriptionally repressive, whereas if you don't have DNA methylation, that's considered transcriptionally permissive. And that's something that I'm going to talk about a lot more in detail. But all of these different layers can be measured site-specifically at high resolution, at unprecedented scales genome-wide. But the one thing that we're lacking are methods that allow us to view each of these layers simultaneously to understand their coordinated dynamics over time. And that's something that my lab focuses on. And in terms of studying genome-wide DNA methylation, people have traditionally used whole genome bisulfite sequencing. And from this type of data, we've really learned a lot. And that is that most of the mammalian genome is highly methylated, with the exception of these regions of the genome that are hypomethylated, as indicated by these blue bars that you're seeing in this methylation track. These regions are these valleys or hypomethylated regions that are scattered throughout the genome, and they typically coincide with gene regulatory elements like enhancers, promoters, and insulators. And by comparing DNA methylation profiles using whole genome bisulfite sequencing, we've been able to understand a lot about the overall dynamics of DNA methylation. And as I said, most of these hypomethylated regions are coinciding with gene regulatory elements like gene enhancers. But if we compare these methylation profiles across different developmental time points and different types of cells from different organ systems, what we can immediately understand is that these hypomethylated regions, or HMRs, can either be highly shared between cell types, or they can be shared between a very specific subset of lineage-specific cell types And some of them are even very cell type specific. And by doing all of these comparisons, we've been able to use this information to trace the developmental history of the cell. But what we really don't understand is how and when these HMRs are established. When do these regions become hypomethylated? How are they demethylated? And how does this relate to other aspects of the epigenome? So models of gene regulation are considered stepwise. This is a stepwise process, hierarchical process that consists of multiple layers of the epigenome. And the idea is that you have enhancers and promoters that are regulating their target genes. Transcription factors are recruited to these sites. They recruit enzymes that reposition nucleosomes around them to allow access to transcription factors. Transcription factors also recruit enzymes that modify histones and demethylases that remove DNA methylation to allow for transcription of target genes. One thing about this model is that it assumes that DNA methylation must be removed for transcription to occur, and it also assumes that changes happen, all of these different changes sort of happen on the same time scale. So what we really want to understand is how are patterns of methylation established in the context of chromatin? So, and what is this relationship over time during a process of dynamic gene regulation? And this all really stemmed from an initial observation where we were looking at DNA's hypersensitivity, which is a measure of chromatin accessibility, and separately looking at DNA methylation genome-wide in human embryonic stem cells. And what we noticed is that many DNase hypersensitive sites are highly methylated. So what that means is that you have this state where a likely gene regulatory element like an enhancer is both open and accessible, but also methylated. And when we look at these same sites in differentiated cells, what we notice is that either these sites get resolved either by closing or by removing methylation. So these types of regions must represent these sort of intermediate states that are representative of sort of a duality of transcriptional regulation. And we felt like if this is true, then we need to be able to capture these changes and how they relate to each other over time, and we really need to understand this better. But the only way we would be able to do this is to observe both changes in chromatin and changes in DNA methylation at the same time in the same DNA fragment population in cells that are undergoing dynamic gene regulation. And so for this purpose, we developed a method that we call ATAC-Me, which is, as it sounds, a method that combines ATAC-seq, which uses a transposase to enrich for open chromatin fragments in intact nuclei that then get subjected to sodium bisulfite conversion, which allows us to distinguish methylated and unmethylated cytosines from the same accessible DNA fragment population. And this is just a picture of Kelly Barnett, who was my first graduate student and the student who also developed this, initially developed this technology. We applied ATAC-Me initially to a system of cell differentiation that involves a cell line they're called THP1s. They're monocytes that can be differentiated into macrophages within 24 hours, and it's a really simple system to do a type of in vitro differentiation. So we applied ATAC-Me over a time course of monocyte to macrophage differentiation of 24 hours. We collected 5 different time points and really densely sampled these time points early on in this cellular differentiation cascade. And right now I'm just showing you the changes in chromatin accessibility for one subset of regions. What we saw is that chromatin accessibility is really robust and transient. And so there's all kinds of different changes in chromatin accessibility that happen across the genome. Some regions open, some regions close, some regions open and close. So I'm just showing you one behavior, and that is a subset of regions that are opening during the time course. But what was immediately obvious to us, and also very surprising, is that throughout this time course, nothing happens to DNA methylation. You have all these changes that are happening to chromatin, but very persistent levels of methylation within these dynamic chromatin accessibility sites. So this was pretty surprising. When we also looked at RNA-seq data from the same cells and the same time points, what we observed is that RNA and transcription follows along very well with chromatin accessibility changes. So regions that are associated with different genes are experiencing the same type of dynamic transcriptional responses. So this was surprising because it implies that first, accessibility precedes DNA methylation changes, and also that transcription is not really dependent on DNA methylation changes. So this kind of was breaking the rules of methylation, that it is controlling when and where transcription happens. And just to point out that when we extended the time course, we didn't observe any changes to DNA methylation until we stimulated the cells with vitamin C, which enhances TED activity, and then we saw a dramatic loss in DNA methylation, but it wasn't necessary to see transcriptional responses. But the one thing that we needed to understand was, one thing I want to point out about these cells is that these are terminally differentiated cells. And so once you stimulate them to differentiate into a macrophage, they no longer replicate. So these are completely post-mitotic cells. And while we and others had observed these similar confusing results where, DNA methylation isn't really doing what we are expecting it to do, we felt like we needed to try to do the same thing, but in a different system where the cells continue to replicate. So are the observations that we made applicable to other differentiation paradigms? So for this, we turn to in vitro differentiation of human embryonic stem cells to neural progenitors. And this was an ideal system to do this because you can do a much longer time course to allow for the cells to transition from an embryonic stem cell to a neural progenitor. It's a very well-established protocol and with very well-known transcription factors that regulate this process. Another thing that's nice about this system is that the cells continue to replicate. And when we sample different time points throughout this differentiation process, we see that the cells are fairly homogeneously transitioning through this differentiation process. So that was one of the reasons, those were several of the reasons that we wanted to use this system to understand chromatin accessibility and DNA methylation relationship. So these are the time points that we actually collected and performed ATAC-me on. So I think it's about 9 different time points from 0 hours to 12 days that we collected. And the first thing that we do when we analyze the ATAC-me data is that we look at the changes in chromatin accessibility. And so from all of the rest of the slides you will see was the work of a very talented graduate student in my lab, Guerin. And so I just wanted to acknowledge her up front because she's done some tremendous work that I'm about to show you. So if we look at just the ATAC part of ATAC-me, at each of these time points, we observe the very dramatic changes in accessibility. What I'm showing you in this heat map is that these regions, it involves 38,000 regions that change over this time course. And you can see that there's many regions that go from blue to yellow. These are regions that are closing. and many regions that go from yellow to blue. These are regions that are opening. Most of these regions are enhancers or repressors. And of course, our goal was to understand what's happening in these dynamic accessibility loci to DNA methylation. And so from sort of this, what I consider to be sort of a 10,000-foot view, we see a couple of different things that are happening. So all of these 38,000 regions that we observe to be dynamic in terms of accessibility, we observed two things with methylation. Number one, the regions that were open initially in this time course are hypomethylated, not surprisingly. But most of those regions remain hypomethylated even after or long after those regions close. And on the other hand, there are a subset of regions that are opening during the time course. Those regions do get demethylated. Many of them do. But the actual dynamic behavior that we're observing is a little bit more complicated just than the opening and closing dynamics that I mentioned. We can actually use computational approaches that allow us to cluster the data into different groups over time. And what we can see is that there are actually very specific, time point specific dynamic accessibility behaviors that we can observe that involve different subset of different regulatory elements. So as I mentioned, there are 38,000 approximately regions that change over time. Most of these regions are enhancers or repressors, and each time point has a unique group of accessible regions. And these regions are enriched for transcription factor motifs that are associated initially with the early time points, pluripotency factors like OCT4 and NANOG, to the more transitional intermediate time points where you see regions that are opening and closing that are enriched for transcription factors like LHX2 and OTX2, and those regions that sort of begin to open at the end of the time course that are enriched for canonical neural progenitor transcription factors like PAX6. Many of these regions that are transitioning from closed to open or open to close, these transient behaviors are associated with substantial switching between different types of regulatory states. Like for the regions that begin as enhancers, those end up becoming another type of regulatory element at the later time points. For example, we have regions that start out as an enhancer and become either quiescent or even a repressor. So we can take basically different types of histone modification data and other types of data and put them together to look at these different types of regulatory annotations. So many of these annotations that we've defined here come from chromHMM. So as I mentioned, using accessibility, we were able to define different types of groups of regions that have different types of temporal accessibility behavior across our time course. And so what we defined were 7 different groups that exhibit dynamic behavior that's unique. And so many of these regions are opening and closing, or going through some kind of transitional behavior. And obviously the goal was to determine what the temporal relationship is between DNA methylation and chromatin changes. And so I don't expect you to look at this and understand it, but the most important thing here is to note that when you look at accessibility, you can see the different types of behaviors that I mentioned before: opening, closing, opening and closing. But DNA methylation seems to be very distinct in terms of its temporal behavior. So just to give you an example, I'm giving you one group of about 5,000 regions that exhibit this type of temporal accessibility behavior where you open and then begin to close by 12 days. So if we look at accessibility, we can see that it starts to change after 12 hours, around 24 to 48 hours, you start to see a very dramatic change in accessibility for these loci. If you look at methylation, though, what you observe is that you do see that these regions get demethylated, but this doesn't happen exactly at the same time as accessibility. This is sort of happening in a delayed process subsequent to changes in accessibility. But we would still consider these changes to be concordant because as regions are opening and becoming transcriptionally permissive, they're also losing DNA methylation. But if we turn our focus to the end of the time course, when these regions begin to close again and actually start to return to their original closed state, those regions continue to lose methylation and remain hypomethylated. So this is just an example of one type of behavior that we observed where DNA methylation dynamics are temporally distinct from chromatin. In fact, if you look at all of these different types of clustered accessibility groups that I mentioned, the main profile behavior of DNA methylation is that these regulatory elements are predominantly demethylated. So here we're just looking at the proportion of regions that either undergo loss of methylation, retain stable methylation, or gain methylation, and their gross behavior over time. And so you can see that even with transient, open, and static loci, you have most of these regions that are undergoing some kind of demethylation. But overall, we observe three major changes. We have regions that are inaccessible and methylated that transition to accessible and hypomethylated, regions that are accessible and hypomethylated that become inaccessible but remain hypomethylated, and then finally these transitory states where you have inaccessible regions that are methylated becoming accessible and hypomethylated, and then inaccessible and hypomethylated. So the major thing that we observe is that these regions, despite opening and closing, are getting demethylated and remaining demethylated. So the next question we had is, what's really driving these different patterns? And I doubt it's very surprising to any of you that we looked at transcription factor dynamics in these regions. The first thing I want to point out is that, as I mentioned, when we take these different groups and we cluster them according to accessibility, each of the groups really is enriched for very, very specific transcription factors over time. And most of these transcription factors we anticipated to be involved in these different time points because they're known transcription factors that are involved in the embryonic stem cell to neural progenitor transition. And one of the benefits of having the ATAC part of the ATAC-Me protocol is that we can use the TM5 cut sites to determine TF footprints within the accessible regions. So if there's a transcription factor bound, this provides a protection against TM5 cutting within the transcription factor bound site, so you can calculate the number of cut sites around these accessible regions to determine the actual footprint that's there, and then you take the sequence motif that's underlying the footprint to determine which is the likely transcription factor that is bound at the footprint. And because we can do that, we can calculate the footprint and we can calculate methylation at the same time, because we also have the methylation state of these DNA fragments. So this is just an example on the right of an LHX footprint at 12 days that's present, and we can determine bound and unbound regions that contain the LHX motif. So we can do this for all of the different transcription factors that are potentially bound in these loci. This is an example of one family of transcription factors. There are multiple members that are differentially expressed across the time course, as indicated in the line plots on the right. And we have, this is a heat map where we've essentially defined each cluster of accessibility regions and then plotted the transcription factor footprint within each cluster, and we can show how these footprints change over time. And if we look at all transcription factor footprints that we are able to determine, and look at transcription factor footprints that are either gained at a specific time point or lost at a specific time point, what we see is that, not surprisingly, when transcription factor footprints are gained at a specific time point, this is followed by loss of methylation. Whereas when a transcription factor footprint is lost at a specific time point, these regions only very slightly gain methylation. In fact, most of these regions remain hypomethylated throughout the remainder of the time course, even after the footprint is lost. So that was very surprising and interesting to us. And it was something that we really wanted to follow up a bit more. We wanted to understand what was happening to the regions that are getting demethylated, and why they are remaining demethylated even after the transcription factors seemingly stop binding and the region loses accessibility. So we know that there are enzymes that are responsible for methylating DNA. These are DNA methyltransferases, and there are enzymes that are responsible for demethylating DNA methylation through hydroxylation of the 5-methylcytosine. And so one of the things that we wanted to understand is how does hydroxymethylation look throughout this process? And so initially, we wanted to look at global hydroxymethylation levels to see if we could observe any global dynamic changes to hydroxymethylation. So one of the things that we did, and this was actually pretty difficult at the time, because it's actually not trivial to be able to detect hydroxymethylcytosine. You know, there are antibodies that can distinguish methylcytosine and hydroxymethylcytosine. And so one of the things that we did initially was to look to do an ELISA using antibody for 5-hydroxymethylcytosine for each of the time points that we collected. And the major observation that we made is that we observe a very strong increase in hydroxymethylation that peaks at around 4.5 days, that eventually resolves and goes back to sort of baseline levels at 12 days. So we were kind of surprised by this, given all of the different types of changes that are happening at the different types of regulatory elements that I mentioned that exhibit these different temporal behaviors. So we were kind of surprised to see that we could observe this one peak of 5-hydroxymethylcytosine. And so we really kind of wanted to, so this sort of led us to this problem that we could observe 5-HMC globally, but we didn't know where this was actually happening. So what regions of the genome actually have HMC. And so this is why we turned to doing 6-base sequencing. So what we did is and actually a lot of the data that I'm about to show is very, very new, but we found out that that we could get access to this assay and this protocol that would allow us to distinguish between 5MC and 5HMC. And so what this protocol does is it takes advantage of a different type of methylation sequencing, which is the enzymatic methylation sequencing. And it allows you to distinguish, as I said, between methylcytosine and hydroxymethylcytosine. So the way that it's able to do this is by taking advantage of different enzymes that can protect hydroxymethylcytosine in the DNA and also copy the methylcytosine to subsequent strands during the assay, during the protocol. So what you end up with is a library of DNA that preserves both the hydroxymethylation and methylated cytosines during the deamination process. And so this protocol, I will say, is very different from whole genome bisulfite sequencing, which doesn't allow you to be able to distinguish between hydroxymethylcytosine and methylcytosine. So we were able to use this in two ways. The first way that we were able to use this protocol is that we were able to use this as a means to verify the methylation states that were called in the ATAC-Me data. And so just to show you what that looks like, what I'm showing you here is basically I'm showing you the proportion of methylation observed in accessible regions determined by ATAC-Me and bisulfite sequencing. And on the other axis, what I'm showing you is the methylation state of the same CG sites observed by 6-base sequencing. So we're just looking at methylcytosine right now. And what was really nice about this assay is that it allows us to see that indeed what we were observing in terms of methylation and the distribution of methylation that we observed across cytosines, CG sites in our ATAC-Me data could be validated by this approach. So we saw very nice concordance between the methylation calls from the 6-base data and the methylation calls from the ATAC-Me data. Another thing we were interested in looking at is whether or not looking at methylation using the 6-base approach, which again is the whole, looking at methylation across the whole genome, and how that related to the methylation dynamics that we were observing using our ATAC-Me approach. So we collected 4 different time points for to do the 6-base sequencing that represented sort of the beginning, the intermediate, and the end of the differentiation protocol that we had performed for ATAC-Me. And overall, when we look at the different groups of accessibility regions that change, that we defined with our ATAC-Me data, what we observe in terms of overall methylcytosine dynamics is that those dynamics track very, very well with one another for the corresponding time points. So in regions where accessibility is closing, you don't really see a lot of changes in methylation that happen. In the intermediate or transient regions where you observe opening and closing, again, we see that in the 6-base data that these regions are losing methylation and remaining hypomethylated. And in the opening regions, we also see that these regions are losing methylation and becoming highly hypomethylated by the end of the time course. So just to bring us back to the initial ELISA that we had performed, when we quantified global HMC levels, we observed that we have this peak of hydroxymethylation that happens right around 4 days. So we had taken, for 6-base sequencing, we had performed 0, 4, and 8 days to profile using the 6-base protocol. And we were able to validate this global change in hydroxymethylation when we look specifically at hydroxymethylation. We can see that on the right-hand side, I'm showing you the distribution of hydroxymethylation levels at each of these different time points, where we can see a change that happens at 4 days, representing the most hydroxymethylation that we can observe over these 3 different time points. And taking this into a bit more detail, I told you that we defined these different accessibility behaviors, and we clustered all of these regions into different groups according to their temporal behavior. So one question we had is, from the 6-base data, if we only look at hydroxymethylation within these different clusters of regions, what do the dynamics look like? And so there were 7 different clusters that we defined, and then an additional group of regions that we're calling static. These are accessible regions that remain accessible and don't really change in terms of their accessibility from 0 to 12 hours over the entire time course. And but if you look at the distribution of hydroxymethylcytosines for each of these different groups, what you'll see is that within the closing regions, we see a mostly stable, although slightly decreasing hydroxymethylation state. In the transient groups, again, we see these regions that are opening and closing. They are also gaining hydroxymethylation and losing hydroxymethylation. And the regions that are opening are also gaining hydroxymethylation. And it really depends, and this sort of varies depending on the time point when the region actually is becoming open. So the overall message here is that according to our 6-base data, the 5-hydroxymethylcytosine dynamics really mirror accessibility dynamics. And this was very exciting for us to see, because what we were seeing with our overall methylation dynamics is that these 2 different layers of the epigenome are quite distinct in terms of how they are changing over time. And ultimately, what that means for the regions that are becoming established during this process. This is just another way of looking at the data that I just showed you, except here, what I'm doing is I'm showing you dividing the hydroxymethylation levels into high and low, where we're really just taking the distribution of HMC, which happens to be very low. I mean, the amount of HMC that we're observing at any given cytosine is pretty in the range of about 1 to 5%, but it can go all the way up above 25%. And if we take those extreme highs of hydroxymethylcytosine content and we ask which regions are displaying the most hydroxymethylcytosine, we see that the very specifically those regions that we defined as with very specific accessibility behaviors display the most hydroxymethylcytosine content. So those are the regions that are either transiently opening or opening, which is maybe what we might have expected for regions that are supposed to be becoming demethylated. And this also seems to be happening predominantly at 4 days. And I just realized that you might not have seen that slide, but hopefully you did. Okay, so I just have a few more slides left before taking some questions. So take-home message here is that now that we're able to really site-specifically look at where hydroxymethylation is happening and which regions are being affected by this demethylation, we believe that we can start to better understand the dynamic coordination between accessibility changes and methylation changes. So just bringing you back to this one slide that I showed you before, we have the ability to look at, from our ATAC-Me data, we have the ability to determine transcription factor footprints to distinguish different time point behaviors according to the transcription factors that bind them. And we also have the added advantage with ATAC-Me that we can look within those footprints at the methylation state of those sites. So we can integrate that with our hydroxymethylation data to understand how methylation is changing within these footprints over time. And so these are two different heat maps. On the left, this is showing the region methylation for the footprints of the previous slide. So we've divided them into different cluster groups according to their time point behaviors. And immediately what you would notice here is that unlike the footprint behavior, we see this predominantly hypomethylated behavior where as footprints are gained and lost, regions are demethylated and they remain hypomethylated. And it was really nice to be able to observe hydroxymethylation also in these regions where you get a gain in hydroxymethylation that sort of lingers around the regions that are becoming and remain hypomethylated. So this was all really nice to be able to validate the observations that we had made and to potentially further understand what is happening in terms of the kinetics of demethylation at these sites and why, in spite of the fact that many of these regions are losing footprints and becoming closed again, why they might remain hypomethylated. And in the final two slides, I wanted to mention that we have been working with data scientists at Biomodal to understand whether we could use machine learning to use methylation to predict accessibility behaviors. So we've been able to take the data that we've generated with ATAC-Me combined with the data generated using 6-base sequencing to train machine learning models to predict accessibility patterns. And just showing you a really like a snippet of that data just to illustrate the idea that once you add both MC and HMC into the model, you get a much even improved prediction of accessibility to what is observed. And so we're very excited about this because one of the issues with methylation is that I think being able to correlate methylation dynamics with other aspects of gene regulation has been pretty tricky. It really depends on the sites that you're looking at. And so you kind of have to ask the right questions of the data in order to be able to understand how it relates to other aspects of gene regulation. So being able to include both methylation and hydroxymethylation in our models, I think will be really important. Finally, overall, our goal is to sort of build a new model for how DNA methylation can mediate genome function. So I've shown you that in this time course of embryonic stem cell to neuroprogenitor differentiation, we can observe multiple modalities of the epigenome. We observe that accessibility is really robust and transient. It responds very quickly and continuously to changes as cells undergo cell fate transitions. But DNA methylation changes are temporally distinct from chromatin changes. DNA demethylation happens, at least in this system, during a very distinct window of time. And it's predominant, all of these regions are predominantly undergoing loss of methylation and never really regaining methylation. So these previously active and accessible regions remain hypomethylated. And now we can use our 6-base genome data to be able to see how hydroxymethylation fits into this diagram and to be able to understand really the more, get better resolution on the dynamics of methylation as these regions are undergoing demethylation. So with that, I would like to acknowledge my lab. There's been a lot of work that's over the years that have gone into this project. Lindsay Guerin is the graduate student who has generated all of this data and done all of the analysis, most of it at least that I mentioned here. And we have great collaborators, including Biomodal. We've been able to partner with quite a few scientists. They have a great team of scientists that we've been able to work with to better understand and analyze the 6-base data. And they're really interested in seeing how this data can be used to better understand some of the questions that we're asking. And thank you so much for your attention. I'm really happy for the opportunity and happy to take questions.
Dr. Hodges, thank you so much for this excellent presentation. It is now time for our live Q&A portion of our webinar. Now, to our audience, if you haven't submitted any questions yet, please do so now. Excuse me. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So, Dr. Hodges, we have so many great questions already coming in, so I'm just going to dive right in. Starting with this one, what is the major difference between whole genome bisulfite sequence and bimodal sequence?
Major difference. Well, so it has to do with how there are several differences, but the one major difference is that you're not using sodium bisulfite to deaminate the DNA. So it's not as damaging to the DNA as bisulfite sequencing is. And the other major difference is that I won't go into all of the detail, but I definitely would refer you to the publication. But there are several enzymatic steps that include glycosylation and methyltransferase steps. So several enzymatic steps that enable prior to deamination that enable you to distinguish between hydroxymethylation and methylcytosine. So there are several steps that that bisulfite sequencing doesn't have that allow you to be able to do that.
Thank you. Now, in your opinion, what is the most exciting insight developed from the 6-base data in your study?
So as I said, we're still digesting this data because it's literally, I just got it in the last few weeks, and so we're definitely following up on this. But so leading up to the 6-base data that I mentioned in the talk, I mentioned that we have these very specific observations that where we have an obvious discordance between what's happening to methylation and what's happening to accessibility. And the difference here is that the changes that are happening to methylation are happening on sort of a totally different time scale than the changes that are happening to accessibility. So accessibility, like with every single time point that we observe, we can observe a very specific subset of regions that are changing, either opening or closing. And so we can capture these really rapid dynamics. But if we look at methylation, we are not capturing those same time point specific behaviors. So one of our goals was to do as many time points as possible so that we would be able to capture anything transient that's happening both at the level of accessibility and at the level of methylation. But what was surprising to us is that we really didn't see these same types of transient effects that were happening to DNA methylation. And so I think that allowed us to make some pretty compelling hypotheses about what could happen. But having the ability to actually observe hydroxymethylation allows us to pinpoint more specifically when the demethylation of these loci begins to happen, because our bisulfite sequencing data doesn't allow us to distinguish those two things. And so being able to actually observe and have the sensitivity to observe hydroxymethylation allows us to sort of pinpoint those regions that are undergoing demethylation at specific time points. And so I think that was the most revealing thing has been one of the most revealing things about this data so far. But it also very nicely validates some of the observations that we have made.
Thank you. Moving on to our next question. Thanks for a super presentation. I'm curious whether...
Christy, I can't hear you.
Oh, well, let's get you reconnected. That's... Okay, how about now? Can you hear me now?
Okay, can you... I'm just not going to use my... Can you hear me?
Yes.
Okay. Not going to use those AirPods anymore.
Got it. Can you hear me okay? Excellent.
Yes.
Okay. Moving on to our next question. Thanks for a super presentation. I'm curious whether methylation and hydroxymethylation typically occurs symmetrically, or whether there are significant instances of hemimethylation.
That's an interesting question that we haven't had a chance to look at in our data yet. Certainly, we're looking primarily at CG sites, symmetric CG sites. But I'm not sure. We haven't looked at that yet, but it's an interesting question.
Very good. Hi, very nice talk. Maybe I have missed it, but is it possible to directly couple 6-base sequencing with ATAC-STARR-seq similar to ATAC-me? Thank you.
This is a great question. Yes, I would love to be able to do that. Certainly, when we started developing ATAC-me, these protocols were under development, and so and the bisulfite sequencing was really the gold standard for doing this. So I would love to be able, now that we can do this, I would love to be able to combine it with ATAC-STARR-seq in the same way that we did bisulfite sequencing. I absolutely believe it's possible, and I think that it might even be better, because for the reason that, as I mentioned, sodium bisulfite treatment of the DNA can be pretty damaging. And so you're already, when you're doing ATAC-Me, you're enriching for DNA fragments from open chromatin, which is a really small fraction of the genome. So you're collecting a small amount of DNA. And so to compensate for that, you're having to use more and more DNA to make sure that, you know, you have enough there to get to the end of the protocol. So I think that if we can employ a similar approach to what we're using with the biomodal assay, then that would be great.
Thank you. We have time for just to squeeze in a few more questions. Thank you for the nice talk. Could you please comment by which mechanism the hypomethylated regions become closed?
Yeah, so I mean, these regions are a lot of, but let's just take a couple of the clusters that we observed to be closing. Many of them are regions that were once bound or initially bound by pluripotency factors. So a lot of these regions might be enhancers in the embryonic stem cells that eventually get closed or repressed when the cells transition to neural progenitors. So they're no longer being actively engaged by transcription factors. So that would be my guess. But the real question I have is, why do they stay hypomethylated? And you know, we're not the only lab to observe these patterns. Even just looking at whole genome methylation data, comparing different cell types, you can see that regions that are established in embryonic cells regions that are established as hypomethylated can remain hypomethylated in differentiated cells. So I think that's a really interesting question, something we were trying to understand.
Excellent. And we have time for one more question. How has the 6-base data improved your understanding of the relationship between epigenetics and gene expression?
Yeah, so this is really the ultimate goal. Because you know, one thing that we understand is that methylation is important. If you disrupt the mechanisms that do methylation or do the demethylation, then you get really unwanted consequences to cell differentiation. Cells don't differentiate. You get genome maintenance problems. So there's just a lot of problems that result if DNA methylation mechanisms are disrupted. So I think understanding the temporal dynamics of methylation is going to be key to understanding why disruption to DNA methylation results in these types of blockades to cellular differentiation. And I also think that what it's going to help us understand about transcription, of the relationship between epigenetics and gene expression, is that, you know, for the last 50 years of studying DNA methylation across many labs, one thing that we can say is that, you know, it's very difficult to correlate methylation changes with gene expression changes, and most of the dynamic is happening to distal regulatory elements. Methylation doesn't change a lot at promoters, so the correlation between promoter methylation and expression has been is not really it doesn't really tell you much or predict about what's going to happen to gene expression if you look at promoter methylation. So I think all of these things are going to help us better understand how methylation is affecting gene expression, and maybe it's telling us that, you know, while DNA methylation may not be directing the immediate changes that are required for transcriptional activation or deactivation, the long-term methylation regulation is what is helping to establish the stability of a cell identity through stable gene expression programs.
Dr. Hodges, thank you for this excellent Q&A session. Do you have any final comments for our audience?
Just wanted to say thank you for attending and listening to me for the last hour, and I'm happy to address any questions you might have. Feel free to email me anytime. Thank you. Thanks, Christy, for moderating.
Absolutely, Dr. Hodges, thank you. And I do want to inform our audience, we did have quite a few questions that we were unable to answer due to time. So those questions, and additionally, any questions you may have during the on-demand period, you can submit those and they will be answered via the email address that you provided at the time of registration. So once again, a huge thank you to Dr. Emily Hodges for her time today and for her important research. And we do want to thank LabRoots and our sponsor, Biomodal, for underwriting today's educational webcast. I want to remind you again, any questions that you have, don't hesitate to submit them. We'll answer them via email. Now, today's webcast can be viewed on demand, and LabRoots will alert you via email when today's presentation is available for replay. We encourage you to share that with your friends and colleagues who may have missed today's live event. To all our friends out there, thank you for joining us. Again, one more huge thank you to Dr. Emily Hodges for her time today. Have a great day, everyone, and we hope to see you again soon. Bye-bye.